scientific knowledge and help advance the scientific field through intellectual discussion. There are a lot of journal clubs in higher institutional organizations like universities, but there's a lack of journal clubs at the high school level, we would say. So that's why Ram and I founded this with the blessings of Dr. Kothari. So today is our first meeting for CSSS. It serves as sort of a test meeting, so thank you all for coming out. With this meeting, we'll revise our processes and address any issues that come up so that we can prepare for a larger audience of, let's say, 500 to 1,000 people that we're targeting.
So for our initial proposed topics, these are the topics that we think would be most beneficial and are really prevalent. Today, in our first meeting, we're going over hallmarks of cancer, but we intend to, like gene editing, vaccine development, medical hardware, different neurodegenerative diseases, iPSC technology, cardiovascular diseases, biology of behavior, medical software, and organ technology. These are the topics we've come up with so far. If you have any other topics that you're interested in, please let us know and we'll take a look at them and see if we can find someone to be able to speak on that topic.
All right, so for today's meeting, for the first 5 minutes, we're going to introduce the speaker and the topic. And then for 25 minutes, there will be a topic presentation. And there will be a post-presentation Q&A session for 10 minutes. Afterwards, if we so decide, we can have a breakout room discussion for 15 minutes, or we can just go straight to our thoughts and conclusions, just maybe 5 minutes long. So today's topic is hallmarks of cancer.
So as Zavraj briefly mentioned before, Dr. Kothari is our mentor, and he's also today's speaker. Dr. Kothari is a research assistant from research assistant professor in Northwestern's Department of Urology. He received his master's in biotechnology from PT Ravishankar Shukla University and completed his PhD in genetic engineering at Mumbai University. He also received the Young Investor Award from the Prostate Cancer Foundation in 2015 for his work on DNA-dependent protein kinase.
All right, now without further ado, we'll give over the presenting controls to Dr. Kothari and he'll share his presentation.
All right, thank you guys, and it's really exciting to participate in this journal club, which is first in the series, and we hope that we will continue and evolve as we progress on our journey. I thought we have been hearing a lot about cancer nowadays since past 1 decade now. This disease is increasing, and so it would be good to put kind of a very basic introduction of what cancer is and what are the general terminologies used, and what causes cancer and what defines cancer, or what characterizes cancer in a very broad term. If we can depict it to a younger population, it will be not only beneficial for them to understand the disease, but also probably will help invoking their interest in research and medicine related to cancer. So what I'm going to do is in the next 25 minutes or so, I'm going to try and go over some of the hallmarks of cancer in a very broad term, without going in detail or without going in nuances. So give me 1 second. All right. So before we actually get into the intricacies of what cancer is, I thought it would be very nice to go over some of the terminologies that are very routinely used and more often or not are confused. People get confused between these terminologies of which terminology to use when and what does it mean really. So we'll start with some of the very common ones, that's like neoplasm. What is a neoplasm? When we call a tumor, or when we use the term carcinoma, or when do we use the term cancer, whether it is benign or malignant, what exactly all these things mean. So a neoplasm is a type of abnormal and excessive growth. Now, neoplasm, as the word defines, neo means new, and plasia is actually a Greek word, which basically means formation. That was adopted in Latin later on also. So as the word means, neoplasia means new formation, some formation of something new. This is characterized by abnormal growth, which usually end up forming a mass, which may or may not be called a tumor. Okay. So when was it used? The Hippocrates used this term called carcinos and carcinoma. And remember, whenever we use the word oma, it means body. So he saw that, you know, cancer cells were projecting some finger-like projections from them, and it looked under the microscope, or it really looked like crab. And so that's how he came up with the word carcinoma, because it looks like a crab. So the description was named after the crab for very obvious reasons. And then later, Roman physician Celsus translated this Greek term into cancer, which essentially in Latin means crab as well. And then Galen, who again was a Roman physician, he used the word onkos, which in Greek means swelling. So you would probably know that cancer is, you know, one of the first things or first manifestation of a cancerous tissue is inflammation. And so he used this word called onkos to describe tumors. And this is a root word for oncology. You must have heard oncology, or a doctor who practices, who treats cancer is an oncologist. So that's how the word, or the origin of the word is. And so depending on this mass of cells, which we call tumor, whether it is cancerous or not, it is termed as benign or malignant. A benign tumor is a tumor or a mass of cells which stays in the place. Malignant tumor or malignant cancer, whatever you want to say it, is when cells start to leave the original site and go to different destinations or different organs, and then they start, that's called metastasis of the cells. So luckily and unluckily, cancer is a disease of multiple steps. You know, it is not like all of a sudden a cancer will develop. The cancer will develop following certain series of events, and in these series of events, cells will start accumulating more and more aggressive properties, and that's how a normal cell becomes a cancer cell. And broadly, they can be divided into 5 different stages. You know, so first of all, because of some environmental stimulation or some genetic mutation, or for some stimulation in growth factors, or decrease in growth factors, or increase in growth factors, there will be a cell that will be genetically altered. And then this cell will start dividing somewhat more rapidly than the normal cells. And that's where we use the word hyperplasia. Again, plasis means formation, hyper means more. So this cell starts to divide a little more than the normal cells, and that's how it forms a cell, a mass which is proliferating a little faster. At this time, the cells they look normal, they are not cancerous at all, they are just rapidly dividing. Then with this ongoing rapid division, the cells become something called dysplastic cells. This stage is called dysplasia, when the cell starts to change its form. You know, an epithelial cell will start to change a little bit. You can see that, you know, the shape is a little different, but it is still not cancer. It is still a mass that is dividing faster. And then the next stage of that is something called carcinoma in situ, or in simple English, in situ cancer or in situ cancer, where the cells that were dividing faster are now starting to form a mass. But this mass is a localized mass. It is staying in place. And that's why we call it carcinoma in situ. In situ means in place. And once they start accumulating more and more, because of this rapid division, they tend to start accumulate exhibiting more and more mutations. They start to gather more aggressiveness, and that's when it breaks the barrier of local tissue environment and gets inside the blood by invading the surrounding tissue. And when that gets inside the blood takes them to different organs, and at this stage we call it as a malignant tumor, or very popularly known as cancer. So cancer cells invade normal tissues and enter blood and lymph, and then they metastasize to different organs through blood vessels. So the process of cancer cells getting into the blood cell is itself is a multi-step process that probably we will discuss in a little detail later. But essentially they have to something called invade the local tissue matrix to get into the blood vessels. And even getting into the blood vessels is not an easy job, because they have to, they have to squeeze through the blood vessels and get into the blood vessel, and then survive that hostile environment of high flow and whatnot. And they have to go to their distant organs, and then again they have to squeeze through the blood vessel to go up the organ and localize and home that particular organ in a completely different situation, a completely different microenvironment that we will discuss a little later. Now the next question is, what exactly causes cancer? So answer is not very simple as you would assume, but very simplistically speaking, the causes of cancer can range from a single base pair mutation to a rearrangement of a whole genome, of whole chromosome, or a part of a chromosome. So you must have heard terms like hyperploidy or hypoploidy, where the whole chromosome number changes. So both of these can cause cancer. So I will cite just one example of each of these events. For example, there is a gene called RAS. What happens is that in the normal cells, when the cells are in homeostasis, RAS is present in a GDP bound inactive form and a GTP bound active form. And this GDP and GTP cycle keeps the RAS activity in check. Basically, RAS is involved in, broadly speaking, RAS is involved in proliferation of cells. But what happens is that because of some stimulation, they may be environmental or dietary or whatever, one particular amino acid changes from I think it changes from glycine to aspartic acid, which is called G12D, or from glycine to cysteine, which is called G12C at the 12th codon. And that causes this RAS to be constitutively active. So it is no longer responding to the regulation, and it becomes constitutively active. And that results in abnormal proliferation and ultimately development of a cancer. So you would probably know that RAS mutations are driver mutations in pancreatic cancer, which is one of the worst cancer of its sorts, with a 5-year survival rate below 10%. The other extreme of this is instead of one base pair, there is a whole chromosomal arm that gets translocated. And you may have heard something called the chronic myelogenous leukemia or chronic myeloid leukemia, which is caused by translocation of a small part of chromosome 9. And this small part of the chromosome 9 goes and joins on a broken part of chromosome 22 and forms something called Philadelphia chromosome, which essentially ends up in forming a fusion between two genes called BCR, which is breakpoint cluster region, and ABL, which is a tyrosine kinase. And this fusion, gene fusion, basically leads abnormal activity of ABL kinase and results in development of chronic myelogenous leukemia. So you would appreciate the span of wrong things that can go, which will result in formation of a tumor or cancer, from starting from one base pair or one amino acid to a whole chromosomal arm or part of chromosomal arm, or sometimes even the whole chromosome set. So can a heterogeneous disease like cancer, can it be defined by some basic, I would say, on some basic platform? Are there some basic rules that every cancer has to follow? The answer is, if you really dive deep, the answer is no. But yes, on a broader perspective, every cancer has some characteristic that it definitely it shows those characteristics, and they can be divided into six different parts. And this six different parts were excellently reviewed by two of the very famous scientists, Hanahan and Weinberg, and that's what we are going to take help to understand the topic of cancer. We are going to take the help from these reviews. And so what are these six essential alterations that result in formation of tumor or cancer? They are self-sufficiency in growth signaling. Then cancer cells must be insensitive or somehow acquire the ability to evade the anti-growth signals. They have to acquire the ability to invade the tissue and go to different organs or distant organs, something called metastasis, as we discussed earlier. They have to have limitless replicative potential, because the cancer cell will go somewhere in, let's say, in some other organ, and it has to multiply, and it has to, generate its replicas. Well, not really replicas, but it has to generate more number of cells. From where it will get the nutrition we all need nutrition to grow, and so cancer cells will also need nutrition to proliferate and grow. And they have this ability to form new blood vessels. So sustained angiogenesis, that's what is their next hallmark. And then, of course, they have to somehow evade the apoptosis or programmed cell death, so they don't just die, but they keep on proliferating. So in the next few slides, we are going to go over these hallmarks of cancers one by one, not in greater detail, but on a very bird's eye view. But at this point of time, we should know that these six hallmarks are common to most of the cancer types. And you will also appreciate why cancer is a rare disease, because you see, a cancer cell has to acquire these six hallmarks before it goes on and successfully forms a tumor. So obviously, and each of these process is very tightly regulated. So there has to be a multi-step breaching, something like, if there is a cell or a prison where you have to get out, you have to make sure that you are breaching each and every security point in order to go out. And so it's a multi-step process, and that's why it is a rare disease, although we see a lot of cancer cases now. So going for one by one, the first of the hallmarks is self-sufficiency in growth signaling. We know that normal cells require stimulation for proliferation. These stimulations are very tightly regulated by growth factors. These growth factors are usually produced in something called a paracrine mode, where a growth factor is produced at one spot or one organ of a body, and then it acts on another organ of the body, right? For example, you have hypothalamus producing a lot of hormones, and then it works on a different organs of the body. It travels through the body, and so on and so forth. So in normal scenario, the normal cells rely on signals coming from other places. What cancer cells do is they acquire the ability to be self-sufficient in growth signaling. So they ask, why should I depend on other organs to get the growth factors? I'll prepare my own growth factor. So how do they achieve this kind of a self-sufficiency? Many ways, but broadly it can be categorized into three parts. The first one is aberrant ligand-induced activation. So as I said, that normally the way the normal cells function is that they respond to growth factors that are produced in different organs, but they act on different organs. So what cancer cells do is they start producing their own growth factors rather than relying on other cells. So this is the first step by which they tend to acquire self-sufficiency. The second one is the alterations in the receptor. So this itself can be obtained through multiple ways. For example, there can be an overexpression of a particular receptor. For example, in prostate cancer. Prostate cancer is driven by androgen receptor. It's a driver gene known for prostate cancer. What happens is that prostate cancer cells become hyper responsive to low amounts of growth factors. So if the growth factors are low, what they do is they tend to overexpress the receptor for that particular factor on their surface, so that they get more and more growth factors just by funneling more of it into the cell. And so overexpression of growth factor receptor is one way by which they can acquire self-sufficiency. Then there can be mutations. So these mutations can be either in the growth factors or subsequent mutations in the downstream pathways, which result in constitutive activation or independence from the growth factors. A classic example of that is the FGFR mutation, fibroblast growth factor receptor mutations, which starts to act continuously in a switch on position once it starts, when it gets a particular mutation. The third way by which the cancer cells can acquire self-sufficiency in growth signaling is by structural alterations. So there can be, so a receptor can be truncated. If there is a truncated receptor, then it is a constitutively on switch, because a full receptor will respond to the anti-growth signals as well. But if it is truncated, that the part which responds to the anti-growth signals is gone. So now this receptor is constitutively on, and so it will cause more and more proliferation. A classic example is epidermal growth factor receptor, or EGFR receptor. So this is on a cell surface on a paracrine level, and the third level is acquired in the cytoplasmic or a nuclear level, where the receptors will transmit the signals down to the cytoplasm, and then to the nucleus, and it will start transcription of some of the genes. And so the mutations, there can be mutations in any of these pathway members that are involved in transmitting the signals from the receptor to the nucleus. And because of these mutations, there can be aberrant activation of downstream signaling pathways that result in multiple other signaling pathway activation and proliferation of cancer cells. So what happens ultimately, all of these converge into something called a phenotype. And what is the phenotype of cancer cells? They are more proliferative. They have the ability to migrate from one side to the other, or by invading the tissues. They have increased cellular plasticity. They have more aggressiveness, and they are able to evade apoptosis because of either combined or some of these events. So how have we known about all these things into the about cancer cells? Scientists have been growing these cancer cells in Petri plate, something we call as a reductionist view. We have learned a lot about the cancer cells through sequencing and through their phenotypes, through the phenotypic experiments that we keep doing, but we do that in isolation. And we have to remember that cancer cells in body are not found in isolation. They are found in a very dense matrix that involves blood vessels, that involves immune cells, fibroblasts, something collectively we call as tumor microenvironment, which we will be discussing in subsequent slides. But I thought to put this information up front, because we have to understand that whenever we are talking about a cancer cell, it's not just a cancer cell that we are dealing with. We are also dealing with the surrounding stroma, the surrounding environment. And that's why now the research field is shifting itself from a reductionist point of view to a heterotypic cell biology point of view, where the cells are grown in combination with other cells. That's why there are in vivo or mouse models or other models are developed that are either immunodeficient or immunocompetent. Now even humanized mice are coming up to understand how close they can be to the human system, and how a cancer cell behaves when it is found in the body. So this is a very important shift in the research that is taking place nowadays. Now, the second of the hallmark is insensitivity to growth signaling. So we know that we heard about the first hallmark, which is self-sufficiency in growth signaling. Normally, the normal cell not only responds to the growth signal, but it also responds to the anti-growth signals, which tell them, okay, this is the time for you to stop. For example, heart muscles, they do not rapidly divide. For example, neurons, they do not rapidly divide. They have a inherent program that tells them that, okay, this is the time for you to stop and you're no more dividing. But what happens is that, and this whole proliferation is regulated in a very careful manner. Again, you must have heard about a cell cycle, which is divided into 4 major parts: G1, which is growth phase 1, S, which is synthesis, where the DNA synthesis takes place, G2, which is a growth phase 2, and then M, which is mitosis, which itself is divided into 4 different parts. And then through the G1S, G2, and M phase, the cell cycle is very carefully regulated. And when I say carefully regulated, that means it responds to the growth factors and it also responds to anti-growth factors. And that's how it is carefully regulated. So I will take one example of how cancer cells tend to evade this kind of a tight regulation in cell cycle. So there is a gene called retinoblastoma. And this retinoblastoma gene is responsible for the cell to go from G1 to S phase, right? So what happens is that when retinoblastoma is hypophosphorylated, it keeps the E2F transcription factor in check. This E2F transcription factor cannot go and bind to the nucleus and transcribe the genes that are necessary for progression of cells from G1 to S. What happens is when the retinoblastoma is phosphorylated, the phosphorylated retinoblastoma gets degraded and it frees the E2F transcription factor. This E2F transcription factor can then go and transcribe the genes that are responsible for G1S progression. Then there is a gene called TGF-beta, transforming growth factor beta. So this TGF-beta essentially stops this process of phosphorylation of retinoblastoma. Essentially, if you stop the phosphorylation of retinoblastoma, the cell will not be able to progress to the S phase. And that's how it is responding to the anti-growth signal and the cells stop. Now imagine what happens if the cells somehow become unresponsive to TGF-beta. Imagine if there is an inactivating mutation in TGF-beta, what will happen? So it will no more be able to regulate this E2F cycle, and the cells will be continuously replicating because there is no anti-growth factor signaling that is going on in these cells. And so now the next question can be, why does TGF-beta unresponsiveness develop? So common causes are, you can have either the downregulation of receptors of TGF-beta. You can, again, as I explained in my earlier slide, you can have mutant or dysfunctional receptors or faulty transducers. Any one of these events can cause TGF-beta unresponsiveness. And then there is another layer of complexity. TGF-beta is not the only only anti-growth factor that is working to regulate the cell cycle. There are many anti-growth factors. There are many growth factors that are working, that are willing to take through the cell cycle. So this is another layer of complexity, something we call a functional redundancy. That means many proteins can perform similar tasks. So if you block one protein, then the second protein takes over. So that's another layer of complexity right there. As I said, that a cancer cell, when it starts proliferating, it has to make sure, because body has an innate system to kill anything that is going abnormal. That's what you call innate immunity and adaptive immunity. Your immune system keeps everything in check. Your body keeps everything in check. But then if a cancer cell has to survive, it has to make sure that it evades the death, right? That's how it will survive, by evading the death. Now, death of the cell in body happens by two major causes. Either it is a programmed cell death or it is a necrosis, right? And now there is a third kind of a thing coming up nowadays, which is called necroptosis. We won't go into that, but basically two different pathways through which the cell dies. The first one of that, and is a very prevalent one, is a programmed cell death, or also called as apoptosis. The process of apoptosis means elimination of unwanted cells, right? So what happens is when the cells is triggered, that when the system tells it, okay, now you have to die, the cell says, okay, I'll behave myself. It shrinks. Then there is a slow nuclear fragmentation. Apoptotic bodies are formed, and these apoptotic bodies are engulfed by nearby uh, cells by a process called a phagocytosis. And because none of these things are released in the environment, there is no inflammation, because the cells are basically scavenging all the debris of the cells that are dead now. And this whole process is completed within 24 hours, right? So this process is also, just like the cell cycle, is regulated by pro-apoptotic factors and anti-apoptotic factors. So that's how it is regulated. There is a stimulation which will tell the cells you have to die, and then there will be stimulations that will prevent the cells to die if the cells are required. So what are the factors that are that propel a cell to die? They can be DNA damage, they can be genotoxic agents, hypoxia, lack of growth factors, and then there will be anti-growth factors. So for example, if there is an excess of growth factors, then there will be anti-apoptosis. If there is excess of survival signals, then that will be anti-apoptosis. So it is a balance between the pro and anti-apoptotic protein that determines the fate of a cancer cell or a normal cell. So what cancer cells do is they tend to upregulate the anti-apoptotic machinery and downregulate the pro-apoptotic machinery, and that's how they survive, right? The second thing, the second mode of cell death is called necrosis, which is always pro-inflammatory and has a great tumor promoting potential. So unlike apoptosis, where a cell is systematically stepwise degraded, necrosis is simply the cells start disintegrating and starts leaking out its contents into the nearby environment or microenvironment, and that's what causes inflammation. Now, inflammation is associated with the recruitment of a lot of different kinds of cells, including the immune cells, and these cells can be pro-tumorigenic because these cells secrete a lot of cytokines based on the stimulation that they are getting, right? So you must have hurt yourself somewhere, right? Some point in your life when you were playing or something, and you see that you have a wound and then it inflames. Why inflammation happens? Because when inflammation happens, the immune cells and other cells infiltrate the site of wound, and then they try to heal the wound, and by providing new blood vessels and nutrients and all those sort of things. So the timing is important. If it is short while, it is healing, but if it sustains for a very long time, and cancer is chronic, so if it sustains for a very long time, then it can be disease promoting. So the next hallmark of the cancer is limitless replicating potential. So we saw that if a cancer cell has to survive, it has to proliferate faster. And this proliferation can be acquired through either growth signal autonomy, or as we just now said, insensitivity to growth signal or evasion of apoptosis. It can also be acquired by circulating cellular senescence, telomere maintenance, and then there are factors that are yet to be discovered. We don't fully understand how a cell acquires this ability to endlessly proliferate. Now there is a sort of counter notion to endless proliferation as well. So far, whatever we discussed, you may be thinking that, well, if the growth factor signaling is on, or if there are too many growth factors, then the cancer cells will keep proliferating. The more signal, more proliferation. But that's not always the case. A lot of times, excessive amount of some growth factors or some proteins, rather than pushing the cells towards a proliferating state, they can be pushing the cells to a non-proliferating but very viable state. And this state is called senescence. The senescence stage is also sometimes, and not all the times, but sometimes is known as a cause for treatment resistance, because the cells stop proliferating, and majority of the cancer drugs, the way they work, at least earlier days, is by targeting actively proliferating cells, right? And so imagine if a cell is undergone senescence, that means it is not actively proliferating. These drugs probably will not act on those cells which are not proliferating in that effective manner as they will be affecting the cells that are very actively proliferating. Now cancer cells, in order to gain this ability of limitless proliferative potential, they have to acquire certain additional characteristics. For example, they have to break free of loss of contact inhibition. So typically normal cells have loss, or they stop proliferating when they come into contact with another cell, because that tells them, normal cells, that okay, there is no more space to grow. Okay, so you have to stop there. But cancer cells have to, evade this notion. So they have loss of contact inhibition. And how is this loss of contact inhibition or replicative potential achieved? It is achieved by either losing a function of a tumor suppressor. Tumor suppressor by itself means tumor suppressor, a gene which suppresses the formation of tumor. Imagine if the function of this tumor suppressor is lost. So obviously the cells will start proliferating more. Or if there is a gain of function in an oncogene. Oncogene means a gene that promotes oncogenesis. So if there is a gene which is actively or constitutively active, it will be a gain of function, and it will lead to active proliferation or limitless proliferation of cancer cells. Then, as I said, every growing thing needs nutrition. Plants need food, they need soil, they need water, they need sunlight. We need food when we are growing, so does the cancer cell, right? So a cancer cell needs food. How will it get food? It will get food through the blood. So cancer cells have the ability to attract blood vessels. And it's a very simple language when I'm saying attract blood vessels. But it has the ability to attract blood vessels and form new blood vessels, something called as neo means new, angio means blood vessel, genesis means formation. So new blood vessel formation, neoangiogenesis. So they have this ability to promote neoangiogenesis, and through that they get nutrition. Not only nutrition, they also gather their supporting actors that support them in growing and escaping the immune system. These can be regulatory T cells or tumor associated macrophages. It can be a very dense stroma, for example, in pancreatic cancer. One of the reasons why the immunotherapy is very difficult in pancreatic cancer is because the stroma of pancreas is so dense that it is very hard for any cell to actually penetrate that stroma. So the cancer cells have this ability to attract blood vessels, get nutrients, and accumulate the accessory players that help them grow by saving them from the attacks of the immune system. And so angiogenesis itself, again, just like cell cycle or apoptosis, angiogenesis is also very carefully regulated. It is regulated by pro-angiogenic factors and anti-angiogenic factors. And it is again the balance of the angiogenic factors that determines whether the angiogenesis will be on or angiogenesis will be off. And so cancer cells have this ability to turn on the angiogenic switch to gather more and more blood vessels and nutrients and all those things. And then that's how they start metastasizing as well. So the next thing is, well, how exactly do they metastasize? So cancer cells, for cancer cells to metastasize, they have to first of all something called perform local invasion, right? So there is a local tissue that they have to break through. So they invade the local tissues. How do they do that? They secrete something called metalloproteases or other tissue degrading enzymes. So these enzymes start to degrade the local tissue. And that's how a cancer cell reaches a blood vessel. And then it squeezes through the blood vessel. And then through the blood vessel, it goes to an organ. of its choice. So usually every kind of cancer cell has a favorite organ to go, right? And so that's why, because possibly because they get more favorable environment there, right, to proliferate. And once it finds the vicinity of that organ, the cancer cell again has to do this phenomenon of squeezing through the blood vessel and getting into a new organ, right? Something called organ homing. And then it colonizes that particular organ and starts proliferating there. And that's how you see clinically detectable macroscopic metastasis when you see, let's say, a radiogram or a mammogram or something like that. You must have seen some of those things. And this whole process is called invasion metastasis cascade. Obviously, in order to perform all these functions, there are a lot of phenotypical changes that a cancer cell has to undergo, right? And so these phenotypical changes are nowadays they are explored even more, and these are called EMT and MET. The EMT is epithelial mesenchymal transition, and MET is exactly the opposite of that, which is called mesenchymal epithelial transition. So imagine it that it has to, if a cancer cell has to go in the blood vessel, it has to be plastic enough to squeeze and get into the blood vessels, right? And so that's why this EMT is also called plasticity, right? Because it is a transition from the already differentiated epithelial cell, and it is going back to a de-differentiated mesenchymal state, and then it is performing the whole invasion metastasis cascade. And again, this whole EMT or epithelial mesenchymal transition is very, very carefully regulated by hundreds and hundreds of factors. The ones in the red are pro, and the anti, and the ones in the green are pro. And so these factors govern the EMT process, and then subsequently the MET process. So you will again understand that when the cancer cell reaches a vicinity of an organ, it squeezes through the blood vessel again, and then it forms a home in a new organ. And again, there it has to differentiate into from a mesenchymal state to an epithelial state, and then start replicating to form a tumor. So this regulation of EMT and MET nowadays is an area of a very active research. And then this EMT is also regulated by the tissue stroma. That means what is surrounding the tissue, and how the EMT process itself regulates or hijacks the stroma. This whole intricate network and understanding of this network is a very active area of research nowadays. So now the past decade or two has seen tremendous advances in technology, and because of that we are able to understand cancer in deeper and deeper level. And so there are emerging hallmarks. Apart from these 6 hallmarks that we described, there are emerging hallmarks, and then there are characteristics that help cancer cells to attain these hallmarks, right? What are these enabling characteristics? They are tumor promoting inflammation and genomic instability. As I said, as cancer cells proliferate, they start to accumulate mutations. These mutations result in genomic instability. Means in a normal cell, the genome is stable. It is regulated. After every division, there are certain things that are taken care of. But in cancer cells, this stability is lost. So there'll be loss of some sections, there'll be gain of some sections, and this is what will cause problems. And it is kind of an evolutionary process for a cancer cell to adapt to more and more hostile conditions to be able to survive into the organ system. And because of these characteristics, they acquire the hallmarks of avoiding immune destruction, right? So as I said, our immune system in a healthy individual keeps these cancer cells in check. But through these characteristics, cancer cells acquire the ability to avoid the immune system. And not only that, they also hijack the organs or the organ systems and tilt their metabolic regulation in their own favor. So just like a virus goes and attacks a cell and hijacks the cellular machinery to tilt the machinery into its favor. That's how similarly cancer cells also tilt the machinery, metabolic machinery, towards their favor. So this reprogramming of cellular metabolism happens to support the continuous proliferation. And then of course, as I said, the evasion from the immune system. How do these cancer cells evade the immune system? So first of all, where did the idea come from that immune system is an extremely important player in tumor development? So people notice that tumors are grown much easier. It's much easier to grow a tumor in an immunodeficient mice. It's very difficult to grow a tumor in an immunocompetent mice. That means immune system has something to do with the development of a tumor. And then slowly the researchers discovered the role of NK cells, natural killer cells, T cells. There are different types of T cells. Some of them are phagocytic T cells, some of them are helper T cells, and it's their intricate I would say intricate signaling or intricate relationship that determines the fate of a cell, a cancer cell or a normal cell, whether it will be phagocytosed, whether it will be destroyed, not phagocytosed, sorry, whether it will be destroyed or whether it will survive. So something called innate and adaptive immunity that possibly you will learn in coming classes. So as I said in my earlier slide, nowadays the cancer research is not only limited to the cancer cells, but also it takes into account the surrounding tumor stroma, or the tumor microenvironment, as we say. And this tumor microenvironment is a very complicated, very intricate, home or niche for many different kind of cells, including cancer stem cells, immune inflammatory cells, cancer cells themselves, of course, then the blood cells, endothelial cells, fibroblasts, so on and so forth. And what these cells do, all of these cells, they individually are responsible for some or the other function. Some cells will secrete cytokines, some will secrete growth signals, some will bring nutrition, so on and so forth. And each of these function is linked to some or the other outcome in the cancer. For example, prevalence of cancer stem cells is linked to more resistant cancers, to more resistance to chemotherapies. There are certain cancers that are more resistant to chemotherapies, and it is hypothesized that it is because of the cancer stem cells. Well, one of the reasons is the presence of cancer stem cells. As I said, the timing of the inflammation matters a lot. If it's for short time or for a stipulated time, it is healing. But if it's for a long time, then it is disease promoting. Then other cells are the source of growth factors, ligands, signal transducers, as I said, that help the cancer cells to grow and metastasize to different organs. Now, the problem is that you may be thinking, so the cancer cells undergo these changes sequentially. No, they don't. It's not that they will first acquire replicative potential, then the self-sufficiency, then the anti-growth evasion, then the apoptosis evasion. No, that's not the case. They can acquire these characteristics in the way they like. It can be 1, 2, 3, 4, 5, 6 or 1, 6, 5, 3, 2 or whatever sequence they like it. And that's why it is very difficult to pinpoint what exactly is the path that a cancer cell will follow. And that's why cancer has been a very difficult disease to treat or cure. Treatments are available, but cure, when we use the word cure, that's what makes it a little difficult. But knowing problems is good, but do we always focus on problems? No, we are also finding out solutions. Each of these hallmarks and each of these characteristics have been exploited by researchers to develop the counter therapies. So cell cycle, we heard about cell cycle. So there are cyclin dependent kinase inhibitors. We heard about immunity. So then there are CAR T cells now being pushed into the clinical trials. We talked about genomic instability. So telomerase inhibitors are there. We talked about the overexpression or underexpression or mutation or truncation of the growth the receptors. And so corresponding receptor inhibitors are now tested in clinical trials, and many of them are in in the clinics now. We talked about DNA damage. So now the PARP inhibitors are in clinical trials and in the clinic. So every step of these, every characteristic has been used by the researchers to counter the cancer cell and to develop specific therapies against it. Some of them are successful, some of them are being tested, and some of them are being updated as the cancer cells update themselves. And so what we are looking into the near future, we are heavily reliant on the evolving technologies that are helping us to better understand cancer cells. RNA sequencing, whole genome sequencing, methylome sequencing, integration of this data, proteomics. These technologies are enabling us to understand cancer in a better way. And we are now trying to complete the picture of tumor biology by integrating all these omics data. These also help us in early diagnosis. They also help us in identifying robust biomarkers. And the idea of doing all these things is that rather than giving a patient a generic drug, we can give a patient a drug which is more beneficial to this patient based on the patient's genetic data. And so we means the research community is, and the medicine community is now developing these things. And the idea is that soon enough, or very soon, we should be able to deliver the therapeutics specifically to the cancer cells in order to reduce the toxicity associated with the treatment, but enhance the benefit of the treatment in a very targeted manner. And so that was a brief overview of what cancer is, where the near-term research is directed in cancer. And with that, I'm happy to take any questions and get into discussion.
So does anyone have any questions that they want to start off with? I actually have a few, so if you don't mind,
Yeah.
I'll maybe start off. So one thing I was wondering about is that how does age exactly affect the formations of cancer?
How does age exactly affect the formation of cancer?
Yeah.
Many ways. One of the very prevalent one is by down-regulating immune system. So with age, your immune system goes down. And as I said, that immune system takes charge of making sure that your body stays healthy. But with age, the functionality of the immune system is gone, not gone, is reduced. And that's why these cancer cells have better chance to proliferate more. That's only one aspect. By no means I am an expert on aging, but that's only one aspect that I can quickly think of. Then there are many other things. So some of the regulation on a cancer cell may be lost. And then with age also, not only the age, but their dietary habits, their environment, their socio-economic environment, that all affects the cancer cells to develop.
So Dr. Kothari, I actually had a question.
Yeah.
So you mentioned that viruses can cause chronic infection, which can cause cancer. So do you think with this new COVID-19 outbreak, researchers are focused around how that causes interactions with cells that can potentially lead to cancer with this new outbreak?
That's a good question. I don't know at this point in time, because we don't know much about the COVID anyways. But there are proteins that are identified which are required for the coronavirus to infect the cells. And one of the genes that at least I know of is called TMPRSS2, which is also a very, I would say, favorite gene of prostate cancer researchers. And so there certainly are links, but at this point in time, I would spare myself in commenting on whether it can lead to cancer. We don't know. But there are other viruses that lead to cancer, like HPV is one of them. Human papilloma viruses can cause cancer. So there are other types of viruses that cause that cause cancer.
I just had a question, and it's so like I don't know like how much you would know about this, but it's more about other treatments like relating to cancer. Um we know that other treatments exist, obviously, like medicines and stuff to like that claim to fight cancer, I guess. But we see a lot about chemotherapy kind of being advertised as like the golden, I guess, treatment for cancer. But like, would you say that any of these other treatments are, if you want, I can maybe like look up one, but like would you say that these other treatments are as effective as chemotherapy, or do you know of any treatments that are as effective as chemotherapy, or maybe even more effective for fighting against cancer?
I'm not a clinician, so I cannot comment on that. Okay, it would be actually wrong for me to comment on anything on the clinic. So usually what happens is that chemotherapy is a very broad term, right? Chemo means chemical. So any drug, even a targeted drug, can be broadly said as a chemotherapy because it's a chemical, right? But just by the definition, it's a chemical, right? So for example, EGFR inhibitor, it's an inhibitor, which is a very specific inhibitor. It just inhibits EGFR, nothing else, but it's a chemical. So it's a chemotherapy, right? So I know that traditionally chemotherapy word is used as like a generic drug which will affect all proliferating cells, mostly cancer cells, because they are proliferating faster. So first of all, chemotherapy is a very broad term. As far as the other treatments go, at least in the US, as far as my understanding goes, everything has to be FDA approved, right? Whenever you are doing anything with human bodies, it has to be FDA approved, right? And FDA approves the chemicals to be administered in human bodies only when they are extensively characterized. So if something is not extensively characterized, there is no way FDA is going to say, yes, we are going to approve it just because on public knowledge or public wisdom. That's not how it works, at least to the best of my understanding. I may be wrong. As I said, I'm not a clinician. And that's why what you're talking about is old wives' tale. Basically, something that our grandparents or maybe other people have. So like, I started eating raw vegetables and I cured my cancer. Or I don't know how true is that. I don't believe in that because it can be one. And we have to understand, cancer is a very heterogeneous disease, right? Even cancer within a body can be different. It's so heterogeneous. Like the same cell that metastasizes to bone versus what metastasizes to lung will show different characteristics. So cancer is so different within a body that something that can help one person may not help the other person. So how do we know if it is not characterized? What's the underlying cause of the benefit or loss, whatever? So it's very difficult to understand how other treatments, auxiliary treatments, will work on cancer. But one thing we can understand is that, in a very layman terminology, the best thing is to keep your immunity well. So it will save you from a lot of troubles anyways.
Thank you.
Mr. Kothari, I have a question.
Yeah.
I remember you said that cancer, like it comes as a result of genetic mutation, and
yeah,
it could be small or it could be like an entire translation of like a codon
yeah,
or a change in
yeah,
a genome.
Mm-hmm.
So I have a question about, do bigger chromosomal anomalies lead to like a faster development or like a worsened condition in cancer, or is it like all dependent on the different hallmarks that lead to developing?
Yes, it depends. There is no set rule. That's the problem with cancer. As I said, in normal cells, there are set rules. You will follow this. Only if you follow this, you are allowed to proliferate, not in cancer cells. So depending on the ploidy level, or depending on the sort of chromosomal structure alteration that is going on, the cells can survive or can become even more aggressive. It all is context dependent. There is no set rule.
Thank you.
I had a question about the anti-growth factors. So you said that all the cells, I mean, the cells have multiple different anti-growth factors, and kind of like it's redundant. So do all of these anti-growth factors have to be suppressed in order for a cancer cell to be able to like proliferate unlimitedly, or like is there just need to be an imbalance in between growth factors and anti-growth factors?
Yeah, all of the growth factors need to be suppressed is a very hard statement. Not all of them. That's not how it works. Usually what happens is there is something called oncogene dependency or oncogene addiction, right? Every cell, not every cell, a cell is dependent on some oncogenes. That, for example, I quoted an example of pancreatic cancer cell, which is dependent on, not dependent, but KRAS mutations are the driver mutations for pancreatic cells, right? And so sometimes it is the potency of the pro-growth signal that overrides the potency of anti-growth signal. So let's say you may find a scene where all the anti-growth signals are intact, but the pro-growth signal is so strong that it completely overrides the anti-growth signal. Sometimes it's the other way around. Sometimes it's like, okay, there are multiple pro-growth signal, very weak. Individually, they are weak, but there are multiple of them. And so that becomes an overriding force on anti-growth signal. Sometimes the functions of the anti-growth signals are lost. So sometimes you'll find, oh, the oncogenic signaling or the pro signaling is okay, you know, it's normal, but it is because of the loss of the anti-growth signaling. Some of them, because not all of the anti-growth signaling molecules work on a particular stage or a particular cell type. For every cell type, there are different anti-growth signals, and those growth signaling molecules work at different stages of cell cycle, depending on which stage the cell cycle is in. So as I said, it's one of the things that actually is a good thing about cancer cells is that there are, and I say good thing because, you know, it helps people like me to answer in a very vague manner, because there are no set rules. So I can easily escape if I don't, let's say if I don't understand something, I can easily escape by just blaming the cancer cells that they don't follow any rules. So, and that actually is a challenge itself, because yes, I mean, you know, we can assume that yes, there is a anti-growth signaling loss. Okay, what if we fulfill this function? What if we somehow restore the function? Then we will cure the cancer. But that's not the case, because it's not just one thing.
Mr. Kothari, I have another question. It's
Yeah.
about, I think it was like step one, one of the hallmarks, self-sufficiency in growth signals.
Yeah.
I understand like how the receptor sites, they can be changed, or that the cancer cells can make those growth signals themselves, but I don't understand like the third part. I think you said something about like downstream.
Yeah, the effectors. So let me explain that in a little more detail. Okay, so what happens is that, so if you can focus on this particular figure here. So what happens typically that a growth factor binds to a receptor, and then this receptor relays the signal. You know, have you ever run the relay race where you pass the baton to the other person, and then the other person runs, and then he passes the baton to someone else, and then he runs, and that's how you complete the race, right? So the baton is passed basically here. So the signaling is initiated by binding of a growth factor to a receptor, and this receptor then transmits this signal to something called effector molecules, right? So these effector molecules are taking the signal from the receptor to its destination, right? And again, this will be very carefully regulated. Now imagine if there is a mutation in this particular effector here, and this effector starts acting on its own, that it doesn't wait for the signal to come, it will just run. If you are running a race like that, imagine that one of your colleagues just starts to run before you even reach them. and that's how it will start producing wrong things, right? And that's what the activation of downstream signaling is. So if there is a mutation in an effector molecule, it will keep stimulating the downstream molecule, that will keep stimulating the downstream molecule, that will keep stimulating the downstream molecule, and ultimately will result in enhanced transcription of genes that are required for cell proliferation.
Thank you, Dr. Kothari.
Yeah.
So, Dr. Kothari, I just have a clarification question.
Mhm.
So, can trauma advance cancer? So, advance the progression of cancer? Just
Pardon? Stroma what?
Trauma. So, can
Trauma.
cell trauma advance cancer? I'm assuming that based on your presentation, I think it can, but I just wanted to double check.
I have no idea on that. I'm sorry about that. I've never paid attention to that aspect. And again, as I said, I would rather dodge the clinical questions because it is wrong for me to speak anything on clinics or
Gotcha.
diagnosis or treatment.
Thank
I can only answer research related questions, that too in a very specific and a very very localized manner, I would say, because I'm not an expert on all the cancers. I mean, I'm not even expert on prostate cancer. So I don't think anybody can claim himself to be an expert of all the cancer. I don't think so.
So that's actually something I was wondering about. Like, what's the biggest question happening right now in prostate cancer? Like, from what my limited knowledge, I know like there's first initial like general prostate cancer, and then there's a neuroendocrine prostate cancer that more researchers are focusing on right now.
Yeah.
But generally, what's the biggest question that prostate cancer researchers are looking into right now?
Actually, a lot of them.
Okay.
And every lab or every researcher focuses on a different aspect of like, so one of the major questions is how to identify patients that will respond to a particular therapy, right? So for example, androgen receptor directed therapies are prevalent in the space of prostate cancer, but there are some patients that do not respond to the prostate cancer anti-androgen therapies. So how do we identify these patients? What is the biomarker, or if there are a set of biomarkers that we need to identify and characterize that will tell us that, okay, this patient is going to respond and this patient is not going to respond. So that's one of the things. The next question is, as I said, androgen receptor is the driver of prostate cancer. So the question is why androgen receptor is deregulated? What controls the regulation of androgen receptor? How the androgen receptor controls all these different pathways that lead to tumorigenesis, right? So again, two questions. First of all, how androgen receptor is regulated and how is it amplified or mutated? And the second is, once it is done, then how the androgen receptor regulate other things. So that's another thing. And then the third part is, of course, as you said, neuroendocrine. So whether the treatment will lead to the differentiation towards the neuroendocrine phenotype, or whether the neuroendocrine cells are already present in this particular cancer, and then they, over the period of time, they get enriched. So that's another area of investigation. And then there is curative intent, where, okay, now we have so many sequencings done, so many, omics studies are done. And so can we make therapeutic sense out of it? Can we develop new therapeutics that are effective? Can we test them individually, or can we test them in combination with the existing therapies? What are these existing therapies? Androgen receptor directed therapies, or radiation therapies, or chemotherapies, or other therapies like that. And then individual therapies, for example, if you develop a molecule that targets a particular thing, then in which set of patients will this particular drug will be more effective? Again, based on their genetic structure or biomarkers. So those are the active area of investigation nowadays. And then the last but not the least, of course, is to bring the drugs that are used in the later stages and bring them to the initial stages. So, and that is again a very big question, because which drug and in what context will it be taken to the earlier stages itself is a very big question that people are actively investigating. So again, as I said, it's it's such a broad and it's such a heterogeneous and such a complicated disease that at least for a person like me, it would be very unwise to comment on this is the biggest question, because every question becomes big when it is put in the right context, right? So
Awesome. Thank you, Dr. Kothari.
Yep.
Right. Are there any last minute questions? All right. Wonderful, everyone. Thank you, guys. I hope you all enjoyed. After this meeting, we'll send out a Google form just with